is Sage Medic. CEO Chris Apfel is going to present. Chris, take it away.
Thank you very much for having me, and thank you, Jay, for your wonderful presentation. And since you said we should start with a good story, let me start with a question. What would you say if your mother were diagnosed with ovarian cancer tomorrow? What would you do? Would you be aware that with our premise on genomic testing and precision medicine, that in most cases there are no actionable mutations, and that the treatment after surgery would primarily just be chemotherapy with unknown outcomes, often only palliative? The challenge is, in genomic testing, only a minority of patients, according to the Keith Flaherty study from Harvard published in 2020, only 1 out of 4 patients have actionable mutations. And even then, it's not a guarantee that the patient will respond to the therapy. It's a real problem to know who will respond to what therapy. And the others, 3/4, what they usually get is chemotherapy, and that's also hit or miss. And one of the challenges is that we like to have a very reductionistic approach. We just look at the DNA, and we like to believe this tells us everything on how the tumor might respond. But we do know that there are many other things: gene expression, proteomics, metabolomics, cell-cell interactions, because the whole is much more than its parts. And that's the reason why it's so difficult to predict what would work best. The best way to predict it is actually if you expose the cancer to the treatment and understand how the tumor will respond. That would be the real important part. And so that's actually what we developed. We developed a way how we can actually take or get a fresh biopsy taken from a cancer patient, break it up into fragments that have the original cell-cell interaction, tumor microenvironment, etc., and basically replicates what would be in the body ex vivo in the lab. And we can then test dozens of drugs and identify the most effective therapy with over 80% of accuracy in just 7 to 10 days. We believe that this can double a patient's chance to have a tumor response by making sure this patient will not get treated with drugs that we can know ahead of time can't work, and can significantly improve survival, quality of life, and potentially also reduce costs. So this is a real game changer in terms of creating a paradigm shift on how we are going to select the most effective treatment for a cancer patient. And what I've told you is we break it up into fragments. You see here a little image of a tumor fragment that has dead cells, the red ones with the red nuclei, and the green ones are the live cells that are still around there. When we aggregate this into microtumors within 1 day, you can see here a microtumor that's still viable and can grow, although they don't always grow. The key is, can we actually kill it? This is an image where a microtumor was treated, and you can actually see you were able to kill it. Almost all of the cells are dead, and so that's the reason why you say this can work. Of course, it depends on different dosages. You see in this graph here drug concentration, and we know that if you give 10 times the dose that a patient can tolerate, and yet the tumor cells are viable and they're dancing around, they're happy, they're partying, then this cannot work in a patient. And that's actually what we can prevent, that patients get unnecessarily treated with ineffective drugs that would do more harm than good. And then we have on the other spectrum, we have those that are likely to be very, very sensitive, at least a majority of cells. Those can actually be beneficial for the patient, and it makes sense to use those drugs that are more effective for this patient. We have in our panel over 100 drugs. Depending on the tumor type, there are certain guidelines which drugs are on the top priority list, and we can help an oncologist and the patients together to identify the most effective therapy. This is an example of an ovarian cancer patient partly because my mother passed away from ovarian cancer, which also was one of the reasons that made me starting this company. And this patient failed carboplatin paclitaxel. We can see here at the normal 1x standardized dose, there is no effectiveness. PARP inhibitors that cost about $200,000 on average for a patient were actually basically ineffective. And we found that a therapy that's actually rarely used for ovarian cancer, much more commonly used for sarcomas, actually showed really the best effectiveness. This is actually therefore a game changer, and we have made sure that we fulfill all the regulatory criteria. So this does not require FDA approval, but it does require strict regulatory oversight as a laboratory developed test in a California registered CLIA certified lab, and that's what we have obtained. So we also have a CLIA license from the Center for Medicare and Medicaid Services. This is something where oncologists, especially when first and second line treatments have failed, can actually get additional insight into, does it make sense to continue? And if so, what is the most effective therapy for that individual patient? The market undoubtedly is huge. It's a multi-billion-dollar market, even at only $4,800 per test. The serviceable obtainable markets in the US alone is about 500,000 patients, and it's not only applicable for ovarian cancer. It's actually a pan-cancer test that is for all solid tumors, which are over 90% of cancers that patients are dying from today. And we can apply it for patients who are undergoing surgery, where there is tons of tissue that can be shipped to us so that we can actually test it. So no additional biopsy would be needed. And you already have a sensitivity and resistance profile so that you have an understanding what makes sense and what doesn't make sense. And then the other spectrum are those patients who are metastatic, who are recurrent, who have developed multi-drug resistance. There, the big question is there something in the armamentarium that could really make sense and really help this patient? And we can get this from either a core needle biopsy, but we can sometimes also get it from the ascites or pleural effusions. This is undoubtedly a huge market. We have here a very conservative estimation on how much this would cost and what the market penetration would be. And we have modeled this after genomic health market penetration with Oncotype DX. And those exits for precision medicine companies can be in the billion-dollar range. So therefore, this is an investment opportunity that not only can be highly profitable and give you high multiples, it can also make a huge difference to cancer patients. And that's actually what we care most about. And we have, since you mentioned on your slide, you should also emphasize the strengths of the team. I have a medical and scientific background. I've been originally an anesthesiologist and intensivist for over 20 years. I've published over 100 peer-reviewed papers and have done a lot of clinical research and clinical trials, know that field. And when my mother was diagnosed with ovarian cancer and my father had lung cancer, I decided to get my Wharton MBA and become an angel investor. So I'm the chair of life sciences at the Karatsu Forum and in Northern California. And we started this company and have gotten actually these amazing milestones. We are also very proud that we have Dr. Kantz with us, who was the Chief Medical Officer and Chief Scientific Officer of the American Cancer Society. Scott Minnick, who was also Managing Director at ARC Ventures for over 10 years, and Bill Workmeister, who also has a investment banking and venture capital background. And we have, of course, also assembled another scientific and medical advisory board, as well as a strategic advisory board, including Dr. Arkel Giorgio, who was the chief medical officer at United Health, and Mika Newton, who is the CEO of XCures, who is actually a brilliant person who always has amazing ideas, especially when it comes to scaling and market penetration. All right, with that, we have a 2 million bridge node opportunity. That is then followed by a Series A1 round for 8 to 12 million, and that has a projected multiple that includes dilutions of about 520 million, of 12x with about 520 million. So this is basically your opportunity to come in and invest. We are planning to close this by the end of September. So September 30th is what the board suggested for the final day for investments, so that there is also sufficient time to conduct some due diligence. So I'm looking forward to your questions.
Okay, thank you very much. If you can stop sharing for just a minute, I want to see the judges there. All right, let's go with Martina first. Do you have any
Thank you.
questions?
Yeah, thank you for your presentation. Can you please comment on the traction you have been getting?
Sure. So we have launched this assay. Since we launched this by mainly word by mouth, we have generated in the last 12 months about $300,000 in revenue. These were usually out-of-pocket pay. So patients were paying out of pocket usually $5,400.
Laurie, do you have any questions?
Yeah, that was going to be my question too. Um, as far as
Okay.
the market goes, do you have any letters of intent or other formal expressions of interest from insurers or other providers? In other words, do you have a ready pathway once you are ready to introduce this to the market to really start getting it sold and revenue coming in? Thank you.
So we have now received some introductions to a small insurer who is very interested in piloting our solution for a specific set of patients.
Thank you.
All right. Jay, Faye.
Yeah, I'd like to learn more about with the backdrop of, we've looked at companies that are competitive or quasi-competitive to what you're doing. So we know enough to be a little bit dangerous in this area. What is it that you're doing? Because it's clearly a good idea. The question is, what is it that's proprietary to how you're doing it that implies that you will become the category leader in this category?
Yes. So we are the only company that can actually measure cytotoxicity and antiproliferation simultaneously. We actually also now have a recently issued patent on this. And by being able also to include the tumor microenvironment, to have a real 3D assay with a readout within 7 to 10 days, that's actually a very unique combination. The academic spin-outs usually are all the organoid models. That's this NIH-funded drug development route, but it takes you weeks to months to grow it. And very often, especially in low-grade tumors, the tumors don't grow. We actually don't have that problem because we actually just look at the fragments, cultivate that for 4 to 5 days, and then have a readout. So we have a success rate of over 88% and have results within a week or two, as opposed to the organoid models that often have a take rate below 50%, and it takes weeks to months. So
is it the cultivation that you have something that you have your IP around?
The IP around is maximizing the yield of tumor fragments so that we have tissue that we can expose and analyze within just a week.
Thank you.
Okay.
It's one of the many differentiators. There are some other technical things, cytotoxicity and antiproliferation and things like that.
Great.